Right, so let's take a look at the process of mitosis. This is the process that cells use to make exact copies of themselves. So, think about a human. Think about the moment that that human is conceived. So the sperm and the egg have fused together to make one complete cell. Now, before that single cell can become a baby, that single cell needs to copy itself millions and millions and millions of times. And every one of those cells will be genetically identical to every other one, and every one of those cells will be genetically identical to the original cell that was formed at the instant of conception. So that means that every one of those cells will have 46 chromosomes, and all 46 will be exactly the same. Now, that's what we refer to as diploid. So diploid means that the cell has 2 copies of every single gene. All right? 2 copies of every chromosome, one that the individual inherited from its mother and one that it inherited from its father. So in mitosis, the original cell, called the parent cell, it was diploid. It had, in the case of a human, 46 chromosomes. Mitosis is the process by which it divides itself into 2 other cells. Those other cells are called daughter cells, and those daughter cells are also diploid. They also have 46 chromosomes. Now, there are 4 steps in the process of mitosis. Before we walk through the 4 steps of mitosis and make notes, I'd like you to have a look at this animation that has been made, and then come back to this video when you're done. Hopefully, watching this short video will help you to watch the process happening, so that then when you're taking notes, it'll make a little bit more sense. All right, so the first step in mitosis is called prophase. The DNA will have already doubled before this starts, and identical copies of the chromosome will remain joined together. And at that point is called the centromere. So what that means is, I want you to imagine a zipper that unzips from 2 ends. Okay, so you might have a jacket like that. You might have a double zippered jacket, so it can zip downward from the collar, but it can also zip upward from the bottom. So the 2 zippers can meet each other somewhere in the middle of the jacket. That's how DNA opens up as well. It opens up from both ends in toward the middle. So before the first phase of mitosis begins, the DNA has already done that. The zipper has opened up from the top and the bottom in toward the middle, and it's made copies of itself. But that's where it stops. So it stops so that those 2 copies stay joined together. And the reason for that is so that the copies don't get lost in all of the shuffling that needs to happen. So it's imagine making a photocopy of a piece of paper and then keeping the original and the photocopy together with a paper clip. That's the point of not completely separating the 2 strands of DNA. So the DNA molecule opens up from one side and it opens up from the other side, and they copy themselves, but they stay joined together at, a few hundred of those rungs of the ladder. They stay joined together until the very, very last possible moment. And that's so that the identical copies don't get lost. Now, as prophase begins, the DNA coils up on itself, winds up on itself, and becomes visible. And this is when we call the DNA chromosomes. Also, the membrane that forms the nucleus itself, the fence that forms the outside of the nucleus, starts to break down. That's called the nuclear membrane. In the second stage, which is called metaphase, the chromosomes will line up along the equator of the cell, and they'll be stuck to spindle fibers, which are kind of like spider silk. They stretch out from either side. These spindle fibers stretch out from either side of the cell, and they're really, really sticky. And the chromosomes will stick to them at that centromere, at that part that hasn't copied itself yet. All right. Homologous pairs of chromosomes, and we've talked about the word homologous in another video, the homologous pairs of chromosomes will line up along that equator, one underneath the other. Anaphase is where the spindle fibers shorten, and that will pull our double-stranded chromosomes apart at the centromeres. So that exposes those last couple hundred rungs of the ladder that hadn't copied yet. Once they're exposed, their complementary pairs will pop in and complete that copy. And that'll just happen as anaphase proceeds. So in anaphase, those double-stranded chromosomes are being pulled apart, and the chromosomes are getting tugged off to opposite ends of the cell. Finally, the last phase is called telophase, and this is where new membranes form around the chromosome to make two new nuclei, each with the same DNA as the parent cell. And then the cell membrane pinches together so that the cell can be broken in two, so that eventually this will become two copies, identical copies of the original cell. All right, now that you've watched the video once and you've taken notes about all 4 stages of mitosis, watch the video again, the animation again, and see if you can put it all together.